Chapter 2, Biological Classification. Since the dawn of civilization, there has been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom were developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals were easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including those characters like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification system for the living organism have hence undergone several changes over the time. Though plants and animal kingdom has been a constant under all different systems. The understanding of what groups or organisms be included under these kingdoms has been changing. The number and nature of other kingdoms have also been understood differently by different scientists over time. Table 2.1, Characteristic of five kingdoms. Character: five kingdoms: Monera, Protista, Fungi, Plantae, Animalia. Cell type in Monera is prokaryotic, in Protista it is eukaryotic, in Fungi it is eukaryotic, in Plantae it is eukaryotic, Animalia eukaryotic. Cell wall in Monera is non-cellulosic polysaccharide plus amino acids. Cell wall in Protista is present in some. Cell wall in fungi present with chitin. Cell wall in Plantae present cellulosic. In Animalia, cell wall is absent. Monera, nuclear membrane absent, and in rest it is present. Body organization in Monera, cellular. Body organization in Protista, cellular. Body organization in fungi, multicellular or loose tissue. Body organization in Plantae, tissue or organ. Body organization in Animalia, tissue, organ or organ system. Mode of nutrition in Monera: autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic and parasitic. Mode of nutrition in Protista: autotrophic, photosynthetic and heterotrophic. Mode of nutrition in fungi: heterotrophic, saprophytic or parasitic. Mode of nutrition in Plantae: autotrophic, photosynthetic. Mode of nutrition in animals: heterotrophic, holozoic or saprophytic. R. H. Whittaker in 1969 proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain, and thereby a six-kingdom classification. You will learn about this system in detail in higher classes. Let us look at this five-kingdom classification to understand the issues and considerations that influence the classification system. Earlier classification system include bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differ in other characteristics. It brought together the prokaryotic bacteria and a blue-green algae cyanobacteria with the other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. This the classification did not differentiate it between the heterotrophic group fungi and autotrophic green plants, though they show a characteristic differences in their wall composition. The fungi have chitin in their walls, while the green plant has a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista had brought together Chlamydomonas, chlorella, earlier placed in algae within plants, and both having the cell wall, with paramecium and amoeba, which were earlier placed in animal kingdom which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristic and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristic of kingdom Monera Protista fungi of the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapter 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole members of Kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shapes: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrio, and the spiral spirillum. Figure 2.1. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic activity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophic, that is, they depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophils, hot springs, thermoacidophils, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane, biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in a specialized cell called heterocyst. Example, nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrite, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogen causing damage to human beings, crops, farm animals, and pets. Chlorella, typhoid, tetanus, citrus canker are well known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figure 2.3. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasmas are pathogenic in animals and plants. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protistian to one biologist may be a plant to other. In this book, we include chrysophytes, dinoflagellate, euglenoids, slime molds, and protozoan under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotic, the protistian cell wall contain a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by the process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group include diatoms and golden algae desmids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, planktons. Most of them are photosynthetic. In diatoms, the cell wall forms two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are chief producers in the oceans. Dinoflagellate. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending upon the main pigment present in their cells. The cell wall has stiff cellulosic plates on their outer surface. Most of them have two flagella, one lie longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellates, example goniolax, undergo such rapid multiplication that they make the sea appear red, red tides. Toxin released by such large number may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called palicel, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Example, euglena. Figure 2.4b. 2.2.4 Slime molds. Slime molds are saprophytic protists. Their body moves along decaying twig and leaf engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium which grow and spread over several feet. During unfavorable conditions, the plasmodium differ and form the fruiting body bearing spores at their tips. The spores possess true wall. They are extremely resistant and survive for many years even under adverse conditions. The spores are dispersed by air currents. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoan. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The member of this group are either free-living or parasitic. They have flagella. The parasitic form cause diseases such as sleeping sickness. Example, trypanosoma. Ciliated protozoan, they are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia cause the water laden with food to be stirred into the gullet. Example, paramecium. Figure 2.4d. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, a disease which has a staggering effect on human population. 2.3 Kingdom Fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on the moist bread and rotten fruits. The common mushroom you eat and the toadstool are also fungi. White spots seen on mustard leaves are due to the parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing puccinia is an important example. Some are the other sources of antibiotics, example penicillium. Fungi are cosmopolitan and occurs in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. Their body consists of long, cylinder, thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. They are called cenocytic hyphae. Others have septate or cross walls in their hyphae. The cell wall of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from the dead substrate and hence are called saprophytes. Those that depend on living plant and animals are called parasites. They can also live as symbionts in association with algae as lichens and with roots of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore called conidia or sporangiospores or zoospores, and sexual reproduction is by oogoniophore, ascospore, and basidiospore. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second, fusion of two nuclei called karyogamy. Third, meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible matting type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell 2n. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage n plus n, that is 2 nuclei per cell, occur. Such a condition is called a dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reduction division occur, leading to the formation of haploid spores. The morphology of the mycelium, mode of spore formation, and the fruiting bodies form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places, or as obligate parasites on plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospores, motile, or by aplanospores, non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or dissimilar, anisogamous or oogamous. Some common examples are mucor, figure 2.5a, rhizopus, the bread mold mentioned earlier, and albuco, the parasitic fungi on mustard. 2.3.2 Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast, Saccharomyces. They are saprophytic, decomposers, parasitic, or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia, produced exogenously on the special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascus. These asci are arranged in different types of fruiting body called ascocarps. Some example are aspergillus, example figure 2.5b, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacious. 2.3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, bracket fungi, or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, example rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two gametes or somatic cells of different strains or genotype. The resultant structure is dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis takes place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting body called basidiocarps. Some common members are agaricus, mushroom, figure 2.5c, Ustilago, smut, and Puccinia rust fungus. 2.3.4 Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phase of these fungi are known. When the sexual form of these fungi were discovered, they were moved into classes they rightly belongs to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stage of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Collectotrichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic such as insectivorous plant or parasites. Bladderwort and Venus flytrap are example of insectivorous plant and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae include algae, bryophyte, pteridophyte, gymnosperms and angiosperms. Life cycle of plant has two distinct phases, the diploid sporophytic and haploid gametophytic, that alternate with each other. The length of the haploid and diploid phase and whether these phases are free living or dependent on other vary among different groups in plants. This phenomena is called alternation of generation. You will study further details in this kingdom in class 3, in chapter 3. 2.5 Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organism. They are multicellular and their cells lack cell wall. They directly or indirectly depend on plant for food. They digest their food in the internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male or female followed by embryological development. Salient features of various phyla are described in chapter 4. 2.6 Viruses, viroids, prions and lichens. In the five kingdom classification of Whittaker, there is no mention of lichen and some acellular organisms like virus, viroids and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not considered truly living. If we consider living as those animals that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? The name virus, that means venom or poisonous fluid, was given by Dmitry Levinsky, 1892. Recognize certain microbes as causal organisms of the mosaic disease of tobacco. These were found to be smaller than the bacteria because they pass through bacterial-proof filters. M.W. Bejernik 1898 demonstrated that the extract of infected plant of tobacco would cause infection in healthy plant and called the fluid as contagium vivum fluidum, infectious living fluid. W. M. Stanley, 1935, showed that viruses could be crystallized and crystals contain largely of proteins. They are inert outside their specific host cells. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material that could either be RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA viruses. Figure 2.6b. The protein coat called capsid made of small subunits called capsomeres protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in human is also caused by a virus. In plants, the symbiotic the symptoms the in sorry in plants the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, T.O. Danier discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable disease caused by prions are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant CR-Jakob disease (CJD) in humans. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont and the fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they have two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas.